I would like to explore the relationship between your genes, your genome, and your health a little bit. So I would talk about clinical applications with the advanced DNA detection technologies. First, I will introduce a little bit about BGI, then the platforms and our services provided. So very briefly, BGI is the largest genomics research institute in the world, and we have about 4,000 employees with more than 1,500 bioinformaticians. So this makes the data analysis highly effective and there's a lot of expertise in it, and extraordinary supercomputer and cutting-edge cloud computing techniques, and of course a lot of high-quality journals in top-tier publications. So in the past few years, we had about 170 publications in these top-tier journals, such as Nature and Science. So the genome sequencing projects for plants and animals, such as silkworm, chicken, etc., and also for human health and disease. So we found, for example, the gene for adaptation to high altitude by sequencing the Tibetans. So these are some of the licenses and certificates that we have. They are mostly international standards. And these are the branch offices that we have. So we were founded in Beijing in 1999, and then we were relocated to Shenzhen, which is where our headquarters is right now. And then our Asia Pacific Operations Center in Hong Kong, and our Center for Europe in Copenhagen, Denmark, and Center for America in Boston. Yeah, soon. And this is our state-of-the-art clinical diagnostics platform, so including the HiSeq 2000 machines, other sequencers, and also mass spec and real-time PCR. This is our digitalized management system for the lab and also for medical information. And we do all this through our supercomputers and the softwares that we developed by ourselves. So these are all the sequencing, all the platforms based on sequencing for example, agriculture, microbiology, and so on. And today my focus will be on the healthcare platform. So our aim is to achieve scientific discoveries and have these breakthroughs and then apply them onto clinical applications which meet the clinical demand. So our services actually can be separated into these 4 parts. We have oncology, hematology, reproductive health, and also infectious disease. So I will go through each one. For oncology, cancer molecular test, what is available right now is the targeted cancer therapy. So we can see that each of these drugs are specific for each receptors, so such as BCR-ABL, imatinib, and HER2, and so on. So for example, I would talk a little bit about epidermal growth factor receptor. So this is a transmembrane protein, and you can see that when the ligand binds to the EGFR, then the downstream pathway is activated. And this EGFR is actually related to cancer cell survival, proliferation, angiogenesis, metastasis, cell motility, and so on. We found that EGFR is related to a lot of cancers, including colorectal and non-small cell lung cancer. These are the most fatal cancers, and then also in other cancers, such as head and neck, and so on. So this is the EGFR pathway. When, as I said before, when the ligand binds to EGFR, the downstream process is activated. And right now, most of the drug is targeting at either the binding to the EGFR or at the tyrosine kinase inhibitors downstream. So targeting at these two. However, if you have a mutated gene at KRAS or BRAF, for example, then even if you take the drugs that are targeting the other 2 places, this will not be effective for you because you will have it constitutively activated for the whole pathway, even if the drugs inhibit at other places. So that is why targeted drug therapy is quite important. So right now, we have developed these platforms to do targeted cancer therapy gene tests for Iressa, Tarcheva, Erbitux, Vectibix, and Glivec, which is also known as the imatinib. And we do this using the Sanger platform and also the MALDI-TOF. And we are also developing other cancer therapy gene tests. So Sanger sequencing is the golden standard for many years, and this will give us a lot of high full coverage, including all kinds of mutations like insertions, deletions, and so on. And also, we will have very high sensitivity. You can detect at very, like about 1%, like 5% instead of the traditional about 10 to 15% mutation where you can detect. We also use Sequenome's mass array system, which has very high accuracy, more than 99%, full coverage, and also very high sensitivity. And we also have this platform for detecting hereditary breast and ovarian cancer. So this is one example of our joint program with this Saint-Jean Perrin in France, and also BGI for doing all the sequencing, and Sun Yat-sen University, which is the leader of the Chinese Cancer Center. For this one, we see that if you have mutations in BRCA1 or BRCA2 in these two genes, it will be a lot more likely to develop breast cancer. You can see on the figure on the right that if you don't have these gene mutations, then your possibility is only around 2%. But if you do have this mutation, it goes from 50% at age 50 to about 87% at age 70. And this is also related to ovarian cancer. So our strategy is to use targeted capture plus NGS next generation sequencing to capture all of the exons located on BRCA1 and BRCA2 genes. Also for hereditary colorectal cancer, we show that, for example, for one of them, which is familial adenomous polyposis, this is one type of colorectal cancer, and it's affected by the gene called APC. So this APC, if you do have this mutation, then you will get 100% chance of developing colorectal cancer. So it's actually quite important to detect these kind of mutations early in life so that you can take measures to prevent, to treat the disease. And also MMR is causing the other type of the colorectal cancer. For cervical cancer, we see that a rapid increase of cervical cancer cases. So we think it's very important to do also HPV genotyping. This is for the 14 types of high-risk HPV plus the 2 types of low-risk HPV. This is by using our mass spec platform using MALDI-TOF MS and also high throughput sequencing. Up to now, we've accomplished more than 60,000 women for cervical cancer screening tests, and we've also donated about 100,000 tests. Here we see also some gene tests related to hematology. So for HLA genotyping, we are very proud to have very high resolution HLA genotyping for all the loci, and this is by using either Sanger or NGS. Our characteristic is that it's very high throughput, thousands of samples per day, and very low cost compared to the traditional techniques, and also very high accuracy. So first, we'll get the samples and go through labeling, then the laboratory information management system, extract the DNA, amplify PCR, and then construct the libraries, pooling, generate our clusters. Then finally, we'll go to sequencing and then analyze the data. So the report you will get from us is the analyzed data, which is quite easy to read. So for example, we work with the China Marrow Donor Program to screen all of the patients. And also for BGI ourselves, we have some volunteers in this program. So up to now, we've done about 243,000 samples in terms of HLA typing. So you can say that there's a lot of experience for HLA. And in terms of thalassemia, there is, we provide this thalassemia test, which is full coverage, fast, and very accurate at low cost. So for thalassemia, not only detecting the normal strands, but also the deletions that are not so common can be detected. And this is for leukemia fusion gene detection, including BCR-ABL, PML, RARA, and all these different types of fusion gene detections. Now let's have a look into reproductive health. For reproductive health, we have tests that are available at every life stage. So you can actually test yourself before marriage, before pregnancy, prenatal, or for newborn screening. So these are the tests some of the tests that are available. So for example, monogenic disease, you can test pretty much at every life stage. So let's have a look at the NIFTY test, which is a fetal trisomy test. First, I will have a background information about the traditional techniques that are available. So right now in the market, people are usually using the biochemical marker-based tests. So as you can see, the first three are non-invasive based on biochemical tests, but they have quite low detection rates. And then the fourth and the fifth are combined screening tests. So they will give you higher detection rates, but you have to wait until end of week 19 to get to finish the test, and then you get the results even later than that. So the three last ones, these are invasive tests, which can give the fetus a lot of damage and also give you the risk for miscarriage. So in order to avoid all of the drawbacks of the traditional techniques, we proposed a new technique called NIFTY, which is a test with very high detection rate and very low false positive rate. This is a non-invasive fetal trisomy test. So it actually detects all kinds of aneuploidies, all kinds of trisomies. So mainly trisomy 21, which causes Down syndrome, and trisomy 13 and 18. So we use next generation sequencing. We actually only need 5 ml of the maternal peripheral blood. So from this 5 ml of maternal blood, we will do plasma and DNA extraction, and from the DNA, we can from that maternal plasma, we can actually get the fetal DNA. So we can sequence and compare both and find out if the baby has trisomy or not. This is the theoretical basis for NIFTY, because it was found in 1997 that there's much higher amounts of cell-free fetal DNA in the maternal blood rather than the DNA level of fetal cells. So this starts from the 5th week of gestation and disappears after childbirth. So your first pregnancy will not be affecting your second pregnancy. This is the principle of high throughput sequencing, as Abby already introduced. First, there are the 4 bases, and one of them is incorporated onto the template strand. And each one is attached with a fluorescence group, which gives the fluorescence signal, and also the blocker group. The blocker group will make sure that only one base is added in each cycle. So after this base is incorporated onto the strand, it will give off the fluorescent signal, and the high-resolution camera will catch the signal, and then afterwards we can cleave both the blocker group and also the fluorescent group, and the next cycle will continue. This is a simple statistic model to explain this NIFTY test. So in the normal healthy fetus, the amount of amount of DNA genomic units would be about 10 in 200 units, and the mother's DNA would be about 190. But in a baby with trisomy 21, then the amount of genome units would be the same from the mother, which is 190, but from the baby it would be 15 because of the 3 copies. So from this statistically, we can calculate for T-score and L-score and find out the risk for trisomy. So this is a comparison of the other approaches which are quite similar, but we've improved upon this technique and make sure that this is available not only for autosomes, but also available for heterosomes. And up to now, we've done more than 30,000 cases of this and successfully detected trisomy 21, 18, and 13 in all of these samples. And for example, this can be used in cases where trisomy 21 case was missed by the traditional triple test. And then because that one has quite high false positive rate and also it can miss a lot of diagnosis. So if you use the NIFTY test, because its DNA sequencing is much more accurate, and we see that this sample falls into the high risk area. These are some of the papers on NIFTY. So if you're interested, please talk to me afterwards. This is a summary that is a non-invasive trisomy test, which is highly accurate, and turnaround time is only 10 working days. We start at the 12th gestational week, so we get the results at the 14th week. This test has already been in operation in a lot of the countries, such as Korea, Taiwan, India, Israel, and so on, and hopefully very soon in Iraq. And another test that we are offering is the deafness genetic testing for hearing impairment. And this is for neonatal screening. Because for deafness, if you can detect before they start to learn the language, then it's much easier to treat this disease, to have the measures against it. And these are the hearing impairment loci that we test for by using MALDI-TOF. Altogether, these 4 genes and 20 different loci. For using MALDI-TOF MS, we can do multiple loci at one time. So we do 20 loci, and the bottom 2 are the traditional techniques. And the sensitivity is extremely high because there's no fluorescent signal transformation, and we only need 5 working days to find out all about the hearing impairment genes. Other than hearing impairment, these are some of the inherited metabolic disorders that we detect. Altogether, we can detect 35 of them. So this one is also for neonatal screening. So when the baby is born, all you have to do is use the Guthrie card to collect about 3 blood spots. And from within 72 hours after birth. And from these blood spots, we're able to detect all the metabolic diseases and also if the baby has hearing problems. So it will be a little sad to get 3 drops of blood, but after he knows that how much benefit he gets, it's like, yeah, he'll be happy again. And this is for monogenic disease genetics testing. As we know that monogenic diseases are affected by one single gene, one single mutated gene, and this can be a point mutation, frame shift, deletion, or insertion. So in BGI, we've developed a platform to diagnose 153 common monogenic diseases because they are also highly prevalent in the Middle Eastern area. So I actually highly recommend this test. This one is 153 common genes, common monogenic diseases across 16 physiological systems. And for example, like the Gilbert syndrome, cystic fibrosis, and retinitis pigmentosa, and so on. Our approach is the targeted region capture and sequencing. So it's like this man fishing over here on the left, using the traditional techniques. He will be fishing, and if he's lucky, he will get what he's looking for. But our approach is to use a fishnet to grab all of them. So we are sure that it will be lying in there, what we're looking for. And these are the mutation databases that we compare to. First, we screen against the normal person's healthy people database, and then we will screen against the single gene disease databases. So our advantages are that it's high throughput, many samples at once, full coverage, and also very accurate and automated bioinformatics analysis. This monogenic test is recommended for patients with clear phenotypes of some certain types of disease, patients with suspected disease, and also patients that have no phenotypes but they have family history for a certain disease, or also for carrier screening. This can be also used to assist diagnosis and also for research in disease. So what about rare diseases? What about diseases that are not covered in only monogenic diseases? So we know that there are 250 million worldwide people who suffer from rare diseases, and around 80% of these are the ones that have genetic origins. That's why we also introduced another new test that we developed called exome sequencing for inherited disease detection. The monogenic disease just now, we use targeted region sequencing. But for exome sequencing, we sequence the whole exome region. So all of the exons where that is only 1% of the human genome, but that is where most of the human disease is located. So this platform includes monogenic diseases, congenital tumors, and also arrhythmia and pharmacogenetics. And what's more interesting, you can also find out about your personal characteristics, drug response for tumor-targeted therapy, and so on. So for example, for the characteristics that are personal, you can look for something interesting like your response to diet and exercise, your sneeze reflex, also your memory, and how good your eyes would be, your eyesight would be, and if you get flush reaction after drinking alcohol, and so on. Oh, and also resistance to HIV. So for infectious diseases, one of our products would be the HBV drug resistant. So drug resistance. This detects about 11 mutations associated with the drug resistance. So this will give you guidance for the hepatitis B treatment. So we're also developing a lot of other tests and also for research in terms of single cell sequencing, PGD, fetal cells research, and also we're moving towards personalized medicine. So detection trend in BGI, so far in 2011, in 2012, we have a bit more than 1,000 diseases that we can detect, but we predict this number would go up to 7,000 by the time of 2015. We also are do a lot of activities for social responsibility. We gave out about 163 cases, 163,000 cases of free tests for HLA, for HPV, and also NIFTY, thalassemia, and so on. Thank you very much.